So, welcome to lecture number 13. Today, we are going to discuss the generation of diversity of lymphocyte antigen receptor. So, in last class we are discussing like different antibody, different antigen. So, their hypervariable region is different because they need to interact with different antigen molecule, okay, or to be specific different epitopes, right. But the thing is, if you consider, I already told during basic concept discussion of basic concept of immunology, while we are discussing that part, that the number of genes, predicted genes, it is not exactly known, the number of predicted genes in human, say 25,000, 30,000, 35,000 depending on the different algorithms. But if we consider the number of antibody present in our body, say at any time point it is 10 to the power 8 different variety. Okay. So, if each antibody is a protein and each protein should have at least one gene, okay, so then we should have 10 to the power 8 different genes, but we do not have that, right. So, how this diversity is developed? So, that was a big question, though it was solved long time back by Professor Susumu Tonegawa in MIT. Okay, what is the basis, how it is, now we know, but before that, I mean when people was not known what is the origin of this variety, origin of this gene, what we used to, how people know this, how so much variation in the hypervariable region develop or generate in antibody as well as T cell receptor. So, in this in the incoming few lectures, we are going to discuss about generation of lymphocyte antigen receptors. I will mostly focus on the primary immunoglobulin gene rearrangement and their variation, and which is almost equally applicable for T cell receptor gene rearrangement also, but definitely if there is anything special for T cell receptor we will discuss. And as the last part of it, we will see the structural variation in immunoglobulin constant gene, which we already know a little, and how this constant region is changing and how this different immunoglobulin isotypes are formed. So, we will see isotype switching as the last part of it. So, I will start today with immunoglobulin gene rearrangement or generation of antibody diversity. Okay. In one of my basic concept class, I told you to go and check what is the meaning of repertoire. Okay. So, now I am giving the complete collection of antibody specificities available within an individual is known as antibody reparator. So, with time it is changing because B cell is continuously developed. So, their receptor modification and all these things, when you will study the B cell development, you will see it is a continuous process. So, the variety of antibody is also changing, their amount is changing. So, that is why antibody reparator is not fixed, okay, at particular time. Today what I have the variety of antibody in my immune system, it may be different after a month or so. So, this is antibody reparator. So, the complete collection of antibodies specificities available within an individual is known as antibody reparator. But before the genetic engineering or recombinant technology is known or the different techniques were discovered, people used to guess like what could be the origin of this diversity, how come so different type of receptor is generated. So, there are two theories are proposed, one is germline theory, another is somatic diversification theory. In germline theory, it is said that separate gene for each different antibody chain and the antibody reparator is largely inherited. The germline theory is saying that separate genes are there for each antibody chain and antibody reparator is largely inherited. That time the number of gene in human was not known. So, the prediction was not irrational, but which is not true, we know, because we do not have that many genes. And somatic diversification theory is saying limited number of inherited variable region sequence undergo alteration within B cell during lifetime of an individual to generate the observed repertoire. So, now if I ask you which one is true, one is saying each one have gene, each antibody has individual gene or separate gene and it is largely inherited. Another theory is saying that the V region sequence undergo alteration during lifetime and this V region is inherited. Okay. So, let me tell you the answer. The answer is both are partially correct. Okay. Neither one is completely true. It is definite that each antibody is produced by different gene, but how this gene originate it is not saying, and another one, and it is also saying it is inherited. This is also partially correct. Okay. Part of this gene is inherited. So, now we will discuss how this diversity is originate. So, primary immunoglobulin gene rearrangement I will discuss now. Okay. So, this picture you have already seen, right. So, this part is I am coming again and again, you see. So, the red part is the most important region of antigen interaction. This is also known picture, only difference if you see is the leveling of this region, okay, D, E, B, A, G, F, C, C prime and C double prime. Because why this is actually these letters are indicating the different beta strand in this figure. So, this beta strand is designated as B, then E, then B, just to understand between which beta strand this hypervariable region is there. So, if you see that B and C there is one, then G and F there is another, and C prime and C double prime there is another. Okay. And now, this same picture is made little simplified form in here. So, it is A B, then hypervariable 1 and then C. So, between B and C, the hypervariable region between B here and C, if you see this is which one, this hypervariable region is HB1. So, C and C prime. So, this is HB2 and F and G, what is between these two F and G, this one is hypervariable region 3. So, this is a schematic presentation of HB1, HB2 and HB3. Our main idea to see of this this lecture will be just to prove how this variation comes and how this hypervariable region are different. Okay. How come so many variable sequence are present in this total repertoire of antibody. Let us understand first the antibody part and then or the B cell receptor part, then we will see how this is applicable for T cell receptor part. Okay. So, this figure, if you see, this one is again here we are showing. Okay. So, this figure if you go here and just what is the difference here? Difference is HB1 is replaced by CDR1, it is all same. Okay. So, now if you see CDR1 and CDR2 okay, are in this region. So, in the gene I will come slowly. Before that I think we should go here. I will come back to this slide again. So, what is found that in light chain, okay, in light chain the variable region is composed of, in light chain the variable region is composed of two different segment, one is V, another is J, and light chain has only one constant domain. So, there is one constant domain. So, this is the DNA structure, this is the germline DNA or genomic, in the before the B cell mature. So, what happened during maturation or B cell formation? There is a recombination happening. In that recombination, this V and J join together. This is also at the DNA level. In this figure, wherever two lines are there, this is DNA and single line is representing the RNA. Okay. So, these V and J recombine and then make a continuous structure V J together, and which is again there are some space and constant region is there. So, light chain, actually the light chain variable region is present in chromosome or in germline level as two segment, one is V a little bigger, another is J. Okay. So, at germline level they are separated, but by recombination they come together and make the complete variable region. And here L stands for the leader sequence or the signal peptide. In this figure, L stands for leader or the signal peptide. Okay. So, the V and J together makes variable region of light chain. So, I am going back to the previous slide. So, now if you see this V and J, they are separated, they together make the variable region, and if you see that both CDR1 and CDR2, they are already present in variable V segment, but CDR3 is actually contributed both by V segment as well as J segment. And these during this recombination, this part is very important, and we will see with time that means V and C, V and J while joined, they made CDR3, and the CDR3 originates from two or more individual gene segment. In case of light chain, it is two segment, some part contributed by V and some part contributed by J, they become together make the CDR3. Same way, if you see in heavy chain, the variable region, then there is a D segment, D stands for diversity, and a J segment. So, first what happened, D and J join together make DJ, and this is also at DNA level, and then another recombination happened between DJ and V, they together make VDJ, the complete variable region of the heavy chain, and this DJ together, and in fact VDJ all three segment together is making the CDR3. Okay. So, here the CDR3 originates in light chain by two components and in heavy chain by three different segment of DNA. So, the chances of variability in CDR3 is maximum. Okay. So, if you see the light chain now, I am going back and forth. So, so please excuse me. So, from this DNA, say for light chain recombination happened, now it is DNA and it is rearranged. So, VJ in light chain and VDJ in heavy chain rearranged. Then transcription happened. In transcription, we won't see that J was just far in the genome. So, it is together, then by splicing they become together, this is one mRNA, okay, the precursor RNA with the poly A tail, then splicing happen. So, the variable region and constant region comes together. So, variable region and constant region comes together, and which gives the protein product like this. And if you see this region, the antigen binding region, you see the yellow part which is the J region, okay, which is also a part contributing in the CDR3 are very much important, because it is present in the antigen binding site. Same way, if you see in the heavy chain, the DJ comes together make DJ, and then DJ and V together make VDJ, then transcription happen. After transcription, the C region, in this case the heavy chain, you know the C region or the constant domain has, this is, if you consider this is IgG, there are CH1, CH2 and CH3, and this violet color is the hinge region. So, this constant region is composed of CH1 hinge and CH2 CH3, all these are together. So, they come as the single precursor RNA, then splicing happened where VDJ is already joined by recombination. So, VDJ and constant domain come together in the mRNA, and mRNA gives the product of the heavy chain which has variable region which is combined VDJ plus CH1, CH2 and CH3 the heavy chain. But if you see carefully that VDJ all three are taking part in the antigen binding site. So, even they are very small in comparison to the V region, J in case of light chain and DJ in case of heavy chain are very very important, because that makes the antibody different with respect to antigen recognition. Now, we will see how this thing happen. So, so this VDJ recombined, so how this is going to help in the diversity, because this is the basis like what is the construction of heavy chain and light chain gene in the chromosome, but this is not saying anything about the diversity or the so much variability in receptor of antibody B cell receptor and T cell receptor. Okay. So, here is the magic. It was discovered that in our chromosome we have multiple copy of variable region, multiple copy of diversity region, multiple copy of joining region, which actually makes the antibody more specific or different specificity against different antigen, which I do not remember exactly, but I will repeat again. Repeat here if I already told you. There are two type of light chain present. Okay. So, one antibody is composed of one type of heavy chain and one type of light chain. So, there are two type of light chain genes are there, one is called kappa and there is called lambda. So, one set both kappa and lambda has V and J segment separately. So, kappa has some V segment and J segment, lambda has some V segment and J segment. So, what was discovered actually initially when this diversity was discovered at genetic level or molecular level, it was shown that in any individual we have variable region for light chain, say for example, kappa light chain, we have 34 to 38 numbers of V region. For kappa chain joining region we have 5. Okay. And for lambda we have lambda light chain we have 29 to 33 variable region, 4 to 5 joining region. So, total if you consider the light chain it may be, but in one antibody, in any particular antibody the light chain should be either kappa type or lambda type. It is not that in one hand it is kappa and another part is lambda, it is either lambda type or kappa type. Okay. So, it is not that all are present. In heavy chain at the same time the variable region we have 38 to 46 in number. We have a diversity region of 23 or the D segment 23 and joining we have 6. And what does it mean or what is that? If you see in the chromosome level or the if you make a cartoon or the map of the genetic, it is like this. In light chain, okay, this is a lambda light chain locus in chromosome. In lambda light chain locus, it is say there are how many I said? I said 34 to 38 variable region. So, sorry 29 to 33 variable region in case of lambda. In lambda chain we have V lambda 1, V lambda 2, V lambda 3, 4, 5, 6 and up to 30. Okay. So, 1 to 30 and then there is a space, then there is one joining region, constant region, joining region, constant region. How many we have? We have 4 to 5 joining region for lambda. So, similarly, we have say for example, this case we have 4. So, we have 1 J 1 lambda 1 constant 1, J 2 constant 2, J 3 constant 3, J 4 constant 4. So, this all this variable region are in series, then alternate J1 and constant region. In case of kappa, the arrangement is slightly different. In case of kappa, it is all this 34 to 38 variable region again in tandem or inverted way, that I will come what is this inverted and tandem. So, they are present one after another, then there is a space, then all 5 J region is here, then only one constant domain. Okay. So, this is the chromosomal arrangement of kappa light chain. The heavy chain, we have 3 domain, one is B, D and third one is J, right. So, how many variable domain? Say for example, if I consider that in average there are 40 variable domain of H chain, we have 40 variable region, then as space, then all 23 D region is one after another, and then 6 joining region, and then this C mu, mu stands for IgM. Okay. All these you the constant portion, if you remember IgM, IgD, IgG, it is normally the gene is named like IgM is mu, IgD is delta, IgG is gamma, like that, and IgE epsilon, IgA is alpha. So, same way C mu is IgM. Okay. I hope you understand. So, these are the numbers and this is the orientation, clear. So, lambda is this. So, now, so before going to do that. So, now if you think that variable region, say I am considering only say kappa. Okay. I have, for example, I have 30 variable region and 5 joining region. If there is an opportunity or a chance, like any of the variable region can join any of the joining region and finally make the complete variable region of light chain, because this is a segment of variable region of light chain. So, variable segment of kappa is 30 and the joining region is 5. If it is so, so any one of this variable region can join any one of this. So, there are say this is I am talking kappa. So, there are 38 total and there are 5 total joining. So, what will be the number possible? It is very simple mathematics. How many different kind of segment, variable segment we can have? The mathematics is very simple, 38 times 5. If randomly any variable region can join with any joining region, the total number of variability will be 38 into 5. Okay. Same way, if we consider for the lambda chain, here there are 30 variable region and there are 4 joining region. There are 4 joining region. So, what is the possible variation? Again simple mathematics, 30 times for 4 is equal to 120. Okay. So, now there are total number of variation possible here is 30 times 4 is 120 and here 30 say 40 for roughly calculation, say if this is equal to 40 and we have 5, what will be the total? It will be 200. Okay, 200, or to be too specific, it is 190. Okay, 190. So, this is the possibility 120 possible lambda variable region, 190 to 200 possible kappa light region. Same way, if you calculate how many different kind of heavy chain is possible, what we have to do? We have to just multiply this variable segment into, because any D can join with any J. So, that will give actually 23 into 6. So, this is the combination of possible combination of D and J, and then that combination can join any one of these 40. So, this will be total number is 40 into 23 into 6. Okay. So, you can easily calculate. I am not spending time for calculation. Okay. So, now suppose this is x. Okay. Suppose this is x, and so if I consider now, so what is, I am telling you the number again, light chain 120, lambda light chain 120 possible variation, kappa light chain 200 possible variation, and heavy chain variation where randomly any d can join with any j and any dj combination can join with any v segment, the possibility is x. So, that gives some variation. So, we do not have that many gene, but after recombination different segments can produce variety of variable region of the gene segment, and you know that constant region does not have any role in the antigen specificity. So, they do not have many, we have very few, either one is fine, right. It may be lambda or kappa or heavy chain. How many are there? Heavy chain only 5 different are there, A, D, E, A and G. Okay. So, different, there are few subtypes that we will discuss later if when time comes. So, now what is happening in antibody molecule? What is there in antibody? So, let us see. In antibody molecule, what is there? Antibody is the combination of one heavy chain and one light chain, right, which is linked with a disulfide bond. Same thing is repeated here. Okay. One heavy chain, one light chain, and they are also linked. So, in any antibody there should be how many light chain? In any antibody there is one light chain and one heavy chain, and finally, they are dimer. Okay. Heavy chain light chain combination is dimer. So, now if you go back to the calculation, what we see 120, any one of these 120 can make antibody molecule any one of this x, right, any one of this x, and any one of this 200 can make antibody molecule, final antibody molecule with any of this heavy chain. So, what will be the possible number? Any one of 120 and any one of 200. So, total is say 3 this one 120 plus 200 is equal to 320. So, 320 is the possible light chain, and we have x number of possible heavy chain. So, if this is also random, if this is random, like any one of these 320 and any one of this x can recombine, what is the possible number of antibody molecule? Very simple mathematics. Again, you just have to multiply 320 times x, that gives you the variety of different antibody. Even that, I mean, it is surprising, I mean, so few number of segment just by recombination and random recombination can give so many variety of antibody, but if you calculate the number, it is still very very low with respect to the number of antibody we are having at any time point. So, next time we will see, or in the next lecture we will see how that number we can reach. Today we will finish here, and in next class we will see how that 10 to the power 8 or 10 to the power 10 variety or 10 to the power 13 variety of T cell receptor we can have. These will not explain you that much, but even also, I mean, even if it is not taking you that much number or that many number, but it is also very good, like small segments and random recombination can increase the final product in a good, I mean much more in number than it is present. We do not have to, suppose just 320, if I see 320, how many pieces we know? We do not have to have the 320 gene. So, if you just consider the light chain gene, we do not have to have 320 gene. Okay. Very few segment by random recombination can give us 320 possible gene. Okay. So, in next class we will see how the diversity is increased, what are the other factors of diversity of the nucleotide that immunoglobulin receptor. Thank you for today.